Hello, my name is Mollie Uhrig, and today I will be telling you about global run-on sequencing, also known as Gro-Seq. What is Gro-Seq? Well, it is a method used to measure levels of RNA in the cell. Specifically, it is detecting nascent RNA transcripts. There are other methods that can measure the bulk levels of RNA at a steady state, but they are not transcription specific. Gro-Seq specifically looks at transcription-specific RNA activities and does not have interference from other activities such as splicing or degradation. It can even be used to detect enhancer RNA transcripts. Gro-Seq is important because it can provide information about transcription activity. You can map this transcription activity and quantify it within the genome. Using Gro-Seq, you can look at gene expression levels by assessing the levels of mRNA transcripts across different parts of the genome, and recent studies have even used it to find the function of non-coding RNAs. Next, I'll go through the process of how to prepare your sample for Gro-Seq. So first, you have cells, you lyse them and isolate the nuclei. It is very important here to use an RNase inhibitor to keep the integrity of your RNA intact. Next is called the nuclear run-on phase, so you actually label the nascent RNA using BRDU analog. So BRDU is incorporated into actively made DNA or RNA, so this shows nascent RNA or DNA. So specifically here we're looking at RNA, so we will extract the RNA using chloroform and pellet it. Next, you will need to fragment and purify the RNA, and then you can perform immunoprecipitation by using an antibody on your beads that specifically recognizes BRDU. So in this step, you are only going to isolate the RNA which has been labeled with BRDU, so your nascent RNA. You then wash and elute your RNA so that you only have your RNA of interest, the nascent RNA. You don't have any other RNA here. And then at this point you can do end repair, which will allow you to do adaptor ligation. You then use reverse transcriptase to make your complementary DNA or cDNA. And then using a barcoded PCR primer, you can amplify this cDNA, which will then be sequenced. Once you have your sequence, you can map these sequences to the reference genome to see just where in the genome these RNA transcripts are. Using this, you can assess levels of gene expression across the genome. So how does this apply to modules 2 and 3? Well, in Module 2, we talked about genomics and transcriptomics. Gro-Seq obviously has an important sequencing component. You can map nascent transcripts across the genome. You can also map RNA polymerase function across the genome, and you specifically evaluate nascent RNA. As I mentioned earlier, you will not have any interference from other RNA activities such as splicing or degradation. In Module 3, we talked about quantitative analysis of gene expression. So Gro-Seq is very important for this module because we are looking at nascent RNA transcripts and transcription activity, which is directly tied to gene expression. So when you have more mRNA made, more transcripts, you will have higher levels of gene expression in this area. So some limitations for Gro-Seq. Number one, it is limited to cell lines. You must have an exponentially growing cell that is making DNA and transcribing RNA so that you can actually use this label nucleotide, BRDU. If your cell is not replicating and making RNA, this BRDU will not be able to work. It will not label RNA unless it is being made at the time that the label is added. So this would not be able to be used in fixed cells or in some kind of model system that doesn't have replicating cells. So also, Nuclei preparation can introduce artifacts because some of the chemicals used here can be rather harsh. Also, your RNA polymerase can be blocked by damage or secondary structures, so you could have some defect in your signal here if RNA polymerase is not able to actually transcribe and make the mRNA. Also, Gro-Seq requires a known genome sequence for mapping the transcripts to the genome. So if you're working with an organism that does not have a fully mapped genome, this may not be such a good option for you. You wouldn't be able to map these transcripts to the genome to see where in the genome is most transcribed. Also, Gro-Seq only has a resolution of 30 to 50 bases. It is not at the single base level. However, there have been some studies recently that have adapted Gro-Seq to improve this resolution to the single base level. Gro-Seq is very specialized and expensive. At the moment, the best option for this sequencing is through the Illumina sequencing company, and it is very expensive. Some advantages of Gro-Seq over RNA sequencing, which is something that we talked about in class. RNA sequencing measures specifically Gro-Seq sequencing measures specifically the steady state of RNA, whereas RNA sequencing includes information on transcription, processing, and degradation. So RNA sequencing does not specifically look at nascent transcripts. So for this reason, Gro-Seq is a better analysis of RNA polymerase activity and transcription activity of mRNAs specifically. For your controls, you could have a sample without the BRDU label and the nascent RNA, and a sample with beads that do not specifically recognize BRDU. So using this, you can determine the background levels and know what level of your signal is actually representative of nascent RNA. To show you Gro-Seq in real life, we have this paper here, which has actually done a slight variation of Gro sequencing called Gro-Cap, which detects specifically the 5' cap on RNAs. So looking at enhancers and promoters. So here they've done a lot of sequencing, but you can see here Gro-Cap, they have mapped across this gene where are the enhancers and the promoters. So you can see it corresponds to other signals, which they have shown here for histone modifications, all looking at accessibility of the RNA to transcription machinery. So this is useful because it can tell you about where in the genome are enhancers and promoters. Next, this paper looked at nascent RNAs from one gene of interest. So they have induced gene expression of SOD2 gene with an inflammatory stimulus like lipopolysaccharide, and they're wanting to see where in this gene is most transcribed. So here they actually can see that there are some transcripts in the promoter region, some throughout the gene, and some even after termination, which is really interesting. So they see, this is in a leukemia cell line, so they see with this inflammatory stimulus you have higher gene expression because there's a higher amount of RNAs made compared to their control, which was not treated with the inflammatory stimulus. Last, this paper looked at RNA polymerase activity, and so they're using an Arabidopsis model. So they're mapping RNA polymerase, and they see specifically, so they're looking near the transcription start site. So in the sense strand, which is shown in blue, you have pausing just after this transcription start site. And also, there is pausing right after the polyadenylation site, which would be at the end of the transcript. So together they have shown that there's RNA polymerase pausing just after the transcription start site and near the site of termination. So it would be of interest for the RNA polymerase to pause just after the transcription start site to ensure that the cell is prepared for transcribing this gene, transcribing in the DNA into mRNA and actually making this gene, because once the polymerase has started transcription activity, it must finish. It cannot stop and leave halfway through. It must finish through completion. So by assessing the environmental levels at this pausing site, it can see if this transcript and gene actually needs to be made, which can be energetically favorable for the cell. So that is all. I hope you have learned something and enjoyed this presentation on Gro-Seq. Thank you.